Here's our first video on the cell membrane. This one will address, where'd it go? Here we go. Question number 6, which is, can you describe what can move through the integral proteins of the membrane and explain why they can be transported this way? Question 7, can you describe what can move through the phospholipid bilayer of the membrane and explain why they can be transported this way? And 8, can you describe the structure of the fluid mosaic model of the cell membrane? The order of the presentations I'm doing is a little backwards, so this one starts with kind of these final questions, and then the later presentations will have the beginning questions. So feel free to watch them in any order, but I do think that this one, by watching them in this particular order, will make the most sense as far as content goes. Okay, so the first thing is understanding exactly what the cell membrane is, what it is made up of, and how it functions. So as you can see, this would be our example of our cell membrane. And the purpose of the cell membrane is to act as a barrier between the internal part of the cell and the environment. This is going to allow for us to maintain a stable internal environment, basically. The structure of our cell membranes are called a phospholipid bilayer, and the reason it is called a phospholipid bilayer is because it's made up of phosphates and lipids, and if you look here, there's 2 layers of them. So that's where we get this bilayer. So the phosphate part is this top group here, and these bottom tails are our lipids. So we can categorize these as amphipathic molecules. The reason we can consider them that is because they're both hydrophobic and hydrophilic. So hydrophobic is this section down here, our lipid tails, and the reason they're called hydrophobic is because they are water-fearing. We discussed this a little bit in class, this word phobic or phobia, and hydro meaning water, so water-fearing. The top part, our phosphate groups, are hydrophilic, which means that they're water-loving. So that's why the structure is like this. We have our 2 hydrophilic ends on the outside because there's going to be both water internally in the cell and externally, and the hydrophilic portions on the inside away from where the water would be. So here's another image of them, and this is what the phospholipid bilayer looks like. Obviously 3D spread across the entire outside of the cell. Now this is where we want to start thinking about that idea of surface area to volume ratios again, because this is where everything is passing through the cell. So the more surface area to volume ratio we have, the quicker diffusion or osmosis, whatever is occurring, will happen. Okay, here's another image of it. Same thing though, hydrophobic outside, hydrophilic outside, hydrophobic inside. Okay, so this layer, the bilayer here, is shown right there, is basically going to separate the inside of the cell from whatever else is going outside in the external environment. This word cytosol is probably newer to you, but cytosol is part of the cytoplasm. Know that they're very similar, but not the same. This allows this internal part of the cell to have a separation from the surroundings. Without this membrane, there would be no cell because it would all just be the same thing. So you're often going to hear our phospholipid bilayer referred to as the fluid mosaic model. Now, the reason it's called this is because this, the bilayer is actually fluid, and what it means by fluid is not necessarily like water, but it's able to move very easily, which is important for cells because most living things do move, so it's important for our cells to be able to have that kind of a flexibility. So while they are attracted to one another, as you can see here, they're not actually bound to one another. So there is some fluidity to it. The reason it's called mosaic is because if you look at it, it looks kind of like a mosaic. There's a ton of different pieces to it. It's actually rather pretty, but there's all sorts of different carbohydrates and enzymes and cholesterol. There's going to be a lot of different pieces here that I'll talk about in a minute, and that's why they gave it this name, the fluid mosaic model. Oh, and this is really interesting. This is cool. So our phospholipids are actually able to randomly move around, so another reason why it's fluid, and they can even switch places up and down. So the cell membrane also have a diversity of proteins that are embedded in them. These are the membrane proteins, which I just discussed. That's where we get that word mosaic from. Now, all of the combination of all these different membrane proteins allows for basically for movement of molecules or whatever to move inside and outside of the cell. So our membrane proteins can be hydrophilic with a polar side, or they can be hydrophobic with nonpolar side groups. This determines how they'll embed actually in the protein. So if we look here, we have our glycoprotein. This is clearly hydrophilic, whereas internally here we are more than likely hydrophobic. So we have glycoproteins, glycolipids are proteins, lipids with carbohydrates attached. That's the main thing there. The main way to know that is glyco is going to be carbohydrates, and then glycolipid, carbohydrate, fat, carbohydrate, protein. Okay, so here's the first thing that we need to know. We're basically going to go through now at this point a bunch of different ways of things either adhering or entering into our cells. So what's all this stuff now that's embedded in our phospholipid bilayer? So the first thing that we have is a hormone binding site that looks like this. Epinephrine is obviously one of our hormones. And so some proteins that cross the membrane can bind to hormones, which allow the cells to detect external conditions. So for example, insulin, which obviously is related to diabetes. Now this is another interesting one. So we do have cell adhesion, which these adhesion proteins, and basically you just need to know, like if I were to give you the name of this to understand like what their role is. So for hormone binding sites, binding to hormones, obviously. Cell adhesion are adhesion proteins that allow cells to form junctions with one another, so within other cells. Obviously, all of our cells make up all tissue. So without having cell adhesion properties, those cells would not be able to bind closely to one another, and we would not be able to form tissues, and tissues form all of our organs. Next thing we have are immobilized enzymes, which are pictured here. And these are just embedded either side of our phospholipid bilayer, and they'll have their active sites exposed. So these enzymes are crucial in pushing reactions along, speeding them up. We'll talk about enzymes more in the second semester, but there are enzymes embedded in the phospholipid bilayer. We also have proteins in the bilayer that act as basically cell identifiers that transmit information from cell to cell, which is extremely important in our both in our nervous system and in our immune system. Okay, this is a really important topic, passive versus active transport. This is one of your questions. So passive transport is basically an open channel all the time that goes all the way through the phospholipid bilayer, and any sort of small non-charged particle, so it's really important, really small non-charged particles are able to just go ahead and diffuse straight through them. So this requires no energy, it just passes right on through. Whereas our active transport is exactly like what it sounds like. It requires some sort of energy in order for our molecules to come through the membrane. So you can see that there's basically this binding site here. Energy is going to be input, and the protein will open for them to push those molecules through. So it's not as easy as passive transport. Active transport is very specific and requires energy. Kind of, you can think of it as like a pump. this just summarizes what I just talked about. This is a great thing to copy down: hormone binding sites, detect changes in external environment, immobilized enzymes, active sites exposed on inner outer membrane, catalyze reactions. Catalyze means to speed up. Cell adhesion, form junctions, cell-to-cell communication, which is just our cell identifications, passive transport, and active transport. Definitely copy this down. These are really important proteins that are embedded in the cell membrane and our phospholipid bilayer. Okay, so in the phospholipid bilayer, if we look more closely, we have these weird little structures, and that's actually just cholesterol, which is really important, surprisingly, to our functioning phospholipid bilayer. So they're mostly hydrophobic, so they're water-fearing, so they're going to be kind of on this internal portion. Now their job, a very cool job, is to actually help maintain normal conditions in several different environments. So it helps reduce membrane fluidity when it's really warm. So it will actually cause it to be a little bit more structured. But then also when it's too cold, it's going to cause it from getting too firm. So if there's anything that's asking about like what helps maintain the fluid mosaic model during a variety of conditions, it's more than likely going to be the cholesterol. That's its job, is to really help maintain normal conditions. Without this cholesterol embedded, our membrane would actually just break up. It would break apart. Okay, so the reason we have this hydrophilic head is because that will actually make it more difficult for molecules to move through, which is good because we obviously want this the phospholipid bilayer to act as a barrier, to act as a protection between the internal part of the cell and the outside of the cell. I want you to notice here we have a lot of different things that we've already talked about. Lots of integral proteins. We have our cholesterol. We have our glycolipid, which is just a carbohydrate attached to a lipid, and we have our glycoprotein, which is a carbohydrate attached to a protein. Oh, yeah, here it says carbohydrate. So we have our integral protein, our peripheral protein, which is this side piece right here, this extra little protein down here, peripheral on the side, and then these are obviously part of the cytoskeleton. So our phospholipid bilayers are called selectively permeable, which means to permeate something would mean to go through it, but selectively means that some things can go through while others cannot. The reason that it's this way is because of the polarity of the hydrophilic heads. So not everything can just go through. So anything that's non-polar and it's really small, so basically no charge, can just go right on through. However, if it is a polar large molecule, it can't just pass right through. So our phospholipid bilayers are selectively permeable, allowing small non-polar molecules to pass through. Some things that pass through really easily, not surprising, are oxygen, carbon dioxide, and a lot of water molecules. Oxygen, carbon dioxide, that makes sense that it just passes through so easily because we use it so much. Some things that don't just pass through easily though are glucose, other water-soluble molecules, and also ions and water molecules. So as you can see, sometimes water can be either side, but the main thing to understand is that oxygen, carbon dioxide diffuse right through our membrane. So this is an example of a water channel. So water molecules are able to pass through this specific channel called aquaporins, and it allows cells to basically maintain homeostasis, which is, homeostasis, which is basically that maintained internal level of water concentration. You can see that in our water channel that there's both positive charges on those outsides. So what you guys might know now is that while, for example, humans do not have a cell wall, a lot of plants do have cell walls, which is outside of the cell membrane. The reason they have cell walls is because that's actually going to provide structural stability, so it's going to give it some strength, while still being permeable. But it's going to help give it a more rigid structure. That's why it's so necessary for plants to have that. So here's a picture of our plant cell, and the cell walls of the plant cell are going to be very different than the cell membrane. The cell wall is made of cellulose, which is large chains of glucose, and it's going to help, basically you can see here, just help that plant maintain shape. It's really strong. Now, the last thing to talk about are prokaryotic cells. So the whole beginning was our eukaryotic cells, but now our prokaryotic cells are similar but at the same time different. So they're made up of peptidoglycan molecules, and there's two different types of bacteria that we can have for our prokaryotic cells. We can have gram-negative or gram-positive bacteria. So what you want to see here is in our gram-negative, there's actually 2 layers of membrane, whereas our gram-positive is just 1 layer of the membrane, and the peptidoglycan is much larger here. So when we are treating some sort of bacterial infection, it's actually much more difficult to treat gram-negative because there's this double layer, this double membrane. So it's way easier to deal with a gram-positive bacteria than gram-negative due to the structure of their membranes. This is just the last example of what fungal cells are made up of. So fungal cell walls are actually made up of chitin. That's this word right here, chitin, which is basically really long polysaccharides and makes up a lot of exoskeletons. So just so you guys are aware, fungal cell walls are made up of chitin, bacteria made up of peptidoglycan, and plant cells, their cell wall is made up of cellulose. So just know those 3 differences. You might be asked, like, what are each of them made up of? And then lastly, just understanding, let me go back to a good image here, that are eukaryotic cells that have the cell membrane. It's a phospholipid bilayer made with hydrophilic heads, hydrophobic tails, double layer, selectively permeable. That is our main structure of our cell membrane.